Hi, it's Maria.
And Emily.
We are postdocs in the plant science group in the University of Glasgow.
And we had the chance to talk with Wendy Bickmore right before her seminars as part of the seminar series for the Institute. And we talked about a lot of different things with her. I particularly liked her take on how she got into science as a young researcher and then how she became the leader of her own group, as well as now the director of the Human Genomes Unit at the University of Edinburgh. What did you like about our conversation, Maria?
I liked a lot that she explained that how important it is to collaborate with different disciplines, and actually from her own interest in visual aspects and imaging, she has developed these collaborations with people from the optics department. So I think it's something to show that you can expand your horizon.
Yeah, that was a really interesting part of our conversation. So following our introduction here, you can find our complete conversation with Wendy and look back at the Institute website for more information about Wendy Bickmore, as well as our future seminar series speakers.
And in general, please keep checking our Twitter for all the news from the Institute. So hope you enjoy the conversation.
So I thought that we would ask you to quickly introduce yourself and just briefly tell us a little bit about your projects that you're working on now,
okay.
And kind of how you got there.
Okay. So I'm Wendy Bickmore. I run a laboratory at the Medical Research Council's Human Genetics Unit, which is part of the University of Edinburgh. And I'm also now, since August 2015, the director of that institute as well. So I have too many hats. I have lots and lots of hats. I don't know quite how I managed to keep them all on. Scientifically, my interests are in the spatial organisation of the genome, the three-dimensional, and hopefully eventually the four-dimensional organisation of the genome. And I call my subject area the cell biology of the genome. So although I work in a human genetics unit, I really think of myself, as a cell biologist. I love microscopy, I love imaging. I like the power of the visual image.
Yeah.
It really kind of speaks to me more than computer screens
and
black and white gels and stuff like
exactly.
that. So where does that come from? I think that comes from, if you have to look back at what I like doing. When I was a kid, I loved jigsaws. I still do love jigsaws, actually. So I'm obviously, and I love maps, and I love painting and drawing. So I'm obviously, and gardening. So I'm obviously, fired up by visual cues, I think, and I think that is reflected in the way I choose to do my science. I wouldn't be very happy being a biochemist purifying proteins off a column, I don't think. It wouldn't get me quite as excited as seeing things. So I think that's why I do what I do now. But how did I end up doing this? I think it's a rather kind of random walk, I think. And I know people often want to know, how did you plan your career? And the answer is I didn't. You just kind of do one thing, do what you want, I think is what it would be my message to people. Just follow what you love to do. So
we were actually just talking about that this morning, that I had attended one of the Women in Research workshops last year that they put on relatively frequently here at the university. And the keynote speaker was kind of like yourself, a more recent director of their institution or their department in their institution, and told a story about being asked when she decided she wanted to be a director. And she said, well, when they asked me.
Yes, right.
And I actually found and I was telling Maria that at the time I found that to be such a existential relief for me, because at the time I was thinking, oh, I'm going to be in my mid-30s and I'm still feeling like I'm in training as a postdoc, and when will I ever be a grown-up? When will I ever actually know what I want to do?
But that goes for everybody, even in our age, or for young students that they are starting their PhD, or they are about to decide if they want to go into academia and so on. So this kind of big question, how far can you plan your career? And it's a bit of
and you can't really.
Exactly.
You can plan the next step and see that where that takes you.
But it's good to hear that from somebody that has been through these steps, and but in a random way, but still successful.
I think it's almost. I think if you plan, if I set out as undergraduate, say I'm going to become the director of a research, you would fail presumably, because there isn't a route to doing that. And the same way. When our current director was due to retire a couple of years ago, he said to me 2 years beforehand, he said, you could do this. And for the first year I dismissed it. I said, rubbish, I can't do that. And then he kept saying it, and after a year I thought, actually I think I could do it, and I think I can do it better than someone from outside because they don't understand the place. But it was a 2-year process of going, actually I could do this, and actually I might want to do it. It's a very different thing from running your lab, but I'm glad I did it, even though it's very difficult.
So how did you start making the decision from being an early career scientist, let's say post-PhD, postdoc sort of, to I want to run my own lab and potentially have a group of people that I want to work on?
So as a PhD student, so I worked in a really fantastic small medical research council unit in Edinburgh. It was run by Ed Southern at the time, the inventor of the Southern plot. And it was a fantastic research environment. It was in a porta cabin, 4 groups, tiny, probably 10 years ahead of their time in trying to understand the genome as a whole thing, as chromosomes as an entity. And then I decided that I wanted to do a postdoc in human genetics. So I was very much a kind of chromosome biologist, cell biologist at the time. And then I decided actually I wanted to work more in human genetics, so I kind of stepped sideways for a little bit. So I did a pretty standard postdoc actually, it was before there was a human genome map even. We didn't know where genes were, and my project I was given was to try and find and clone the gene for a particular disease, which I failed at, by the way. But along the way, so I was making maps of the genome where I knew the gene must be, and trying to find where the genes were, and I could see that there was a pattern in my map. This is my visual brain kicking in again. All the genes are stuck down one end, and there was this enormous desert down the other end. I thought, that's interesting, but it wasn't wasn't my project, so it's in the back of my mind. And then I was the victim of, or lucky enough to be caught up in building refurbishments. So our lab had to get dispersed to other bits of the campus, and I got what I think my friends and colleagues thought was the short straw. I got sent to the cytogenetics department. So that was thought to be deeply unfashionable. We were the young kids in molecular biology. We were the cool things, right? And cytogenetics was deeply old-fashioned at the time. Fluorescence microscopy was only just starting to kick in, and people starting to realise the power of imaging. So everyone thought, going to a backwater. But actually, so I moved there, and then one of the technicians in the lab there, whose job it was every day basically to sit down and make chromosomes from patients and try and see what was wrong by just studying the pattern. She said, "Have you ever seen chromosomes under the microscope?" And I said, "No." So we made some, and she showed me them. We banded them, and you get this beautiful black and white banding pattern on the chromosomes. And I was just like, oh my god, these are beautiful. And they've got this pattern on them. Oh, and actually my data, I've got this pattern on my data, and I can see this pattern on the chromosome. And it was just one of those eureka moments. Are the two things related? Are what you see down the microscope as a chromosome related to a pattern that's written into the DNA sequence? And so the director of the institute at that time, who was himself a cytogeneticist, was obviously interested to have a young molecular biologist suddenly interested in this area. So it's this juxtaposing of a rather old-fashioned field of research with modern molecular biology, bringing the two together. And he said, first of all, he pointed me in the direction of a few papers coming out in the literature and said, why don't you read these? I think you'll find them interesting, which I did. And he made me give a journal club on one of them. And this paper had taken two of the major classes of repeat sequences from the human genome called ALUs and lines, and labelled them with radioactive tritium and hybridised them back to chromosomes on a microscope slide to see where the repeats were. And they could show the different classes of the repeats. One of them was in the black bands, one of them was in the white bands. And although it wasn't fluorescence and fancy microscopy, it was very old-fashioned radioactive microscopy, I could see, therefore, how you might be able to take that and use it to address my question about where genes might be located in particular places on chromosomes. And that idea coincided just at the time when fluorescence took off. So the ability not to image by having to lay down chromosomes on a bit of film emulsion for 3 weeks to get a few dark black and white spots, but actually be able to see things in colour. So I actually came up with the idea to purify all the genes from the human genome and label them with a green paint, and then take the bits of the genome that are not genes and label them with a red paint and hybridize them together onto chromosomes to see if you get yellow or whether you get red and green stripes. So really, really simple experiment. But I was still a postdoc at the time, coming to the end of my postdoc, and so he said, oh, that's a nice idea, why don't you write a fellowship? So he pointed me to something towards called the Lister Institute. It's actually, I'm now a governor of the Lister Institute, but it was one of the few places at the time where you could get independent funding to start your own lab as a junior PI. And I managed to get one, so that's really what launched my independent career. I had a ball. It was me, technician and student for 5 years. That's all it was. Tiny group. Yeah. There's no big groups in those days. We were just the three of us on the lab bench.
Do you think that type of group is sustainable under current research requirements now?
Not long term, but I think in terms of when you start your group, absolutely. So I have a bit of a concern at the moment. I see this amongst my junior colleagues, that when they get the first independent position, the first thing they do is leave the lab bench behind, go sit in an office and write a grant to try and grow a lab of 6, 8 people or more. And I think that's wrong. It's too fast. You can't recruit students and staff of the quality to populate a lab that big. And you're your best postdoc. At that point, you're your best pair of hands. So you should be in the lab. So I'm very much an advocate still of keeping it small.
And when you're a junior staff, do you feel like you have a bit more of a insulation because you are being a bit more supported maybe from your institute? So you have a little more. You have a bit of protection.
Room. Yeah, no, absolutely. And I think universities are very aware of that these days, and kind of giving people a bit of protected research time because it's tough. At the start, you get a fellowship or something 4 or 5 years, and you've got to basically produce a decent paper. But it's just tough. But anyway, the experiment worked beautifully. I was lucky. I was lucky enough to recruit a PhD student who, on paper, would have looked like a failure. He got a 2-2, so he didn't qualify for a regular research council grant. But he was obviously passionate about chromosomes because he'd gone off and worked as a technician in the cytogenetics department for 3 years. So he was kind of mature. So he had just the pair of hands I needed to do that experiment. So between the two of us, we did this beautiful experiment. I'm really proud of it to this day. And I'm going to have it made into a stained glass one day. It's every single human chromosome lined up, the 22 autosomes on the X and Y, and then with our red and green paints, and we could just see straight away. Oh my God! The red and the green are not in the same place. They alternate along the chromosomes. So it clearly is. That banding pattern that cytogeneticists have used for years actually is a reflection of the underlying DNA sequence. So that's what got me started. And from there, so the next experiment, there's clearly a relationship between the primary DNA sequence and the way chromosomes look at the point when they divide in the cell, where you can really see them as individual chromosomes. So the point of the cell cycle called mitosis. But actually mitosis is a bit boring. I mean, there's not much going on in the genome at that point, bar the chromosomes being segregated. what's happening in the nucleus to these different compartments, the red and the green compartment, are they intermingled? But does the nucleus also have some spatial organisation and way of compartmentalising that? So we essentially took the same experiment and applied it not to chromosomes on the slide, but actually to the nucleus, and then started to see, again, really interesting non-random spatial organisation of our genome with the more inactive compartment around the outside and the places where the genes are most active in the inside. So this idea that there's kind of zones in the nucleus where different kinds of reactions might happen.
I'm also really interested in kind of cellular topology and thinking about the cell as something other than kind of a circle within a circle that we see on paper, that there's a three-dimensionality that we really lose when we draw things in a textbook or whatever. And I was actually just talking over dinner a couple of days ago about DNA and how all of the components that interact with DNA, other proteins or RNAs or whatever, have to fit on what we always indicate as just two parallel lines. And thinking about really the spatial and topological requirements that are needed when you start thinking about, one, how much DNA is actually inside a nucleus, how much that increases when you have to deal with replication, and then all of the proteins and other factors that need to find where they need to bind.
How does the transcription factor ever find it?
How does it fit? I mean, to me, it really, in my mind, I just think of my textbook images, and I think that when I have to start scaling it out to a three-dimensionality, it becomes much more complex than just, the polymerase finds the whatever. And
people tend to understand concepts in a linear fashion much easier than going. So probably that's why the textbooks have something very...
I agree, but I do think it's important to remember that a lot of this structure and organisation we see emerges as a consequence of reactions.
Certainly.
It's not necessarily just there and everything, I must go over there and I must go there. It's an emergent property of the cell's function. But I think my view of it is that at the end of the day, it's all biochemistry. In the end of the day, it's all protein-protein interactions and driving reactions to be efficient. And basically, if you can concentrate things that do the same thing in the same place, you raise the local concentration and make everything much more efficient. So I think that's the role of three-dimensional organisation.
But then we have to
drive concentrations.
Yeah, but then we have to use the kind of hand-wavy things are recruited to these areas. But I always wonder, how does that happen? I
don't think we do. So I always, when I'm looking for mechanisms in three-dimensional organisation, I always revert back to thinking about the nucleolus, because it's such a paradigm for a three-dimensional compartment that does a particular job. And the nucleolus forms because of ribosomal gene transcription and processing. It doesn't form and then the ribosomal DNA goes, I better go over there because that's where I belong. These are self-organising structures. So it's actually very, very simple. But it needs principles, and it's driven by affinities between proteins.
Sure,
right. It's all driven by protein-protein affinities.
So once those proteins kind of associate with one another, then it can more and
more
and more.
Absolutely, more and more and more. And you can actually watch that in living cells. You can watch the nucleoli form from the 5 pairs of ribosomal carrying human chromosomes, and they start off as individual chromosomes, pre-nuclear bodies, and then as they gradually diffuse around the nucleus to come together, they will tend to stick together because they have affinity for each other. So gradually you fuse together to make nucleolus.
Yeah, interesting.
Yeah. So it's not magic. It is at the end of the day just basic biochemistry, but with quite profound consequences. For how the cell works, I think. So my current obsession is how long-range enhancers work, how the gene regulatory switches, which are the bits of our genome that control cell and tissue-specific gene expression, how they work, how they find their target genes in three-dimensional space, because so many of them are a ridiculously long way away from
Yeah.
the genes that they have to regulate. A million base pairs or so. I mean, what a crazy way to set up a genome. but that's the way it is. And I just find that idea quite really interesting, how two bits of DNA have to talk to each other in some way, whether it's through a physical contact and some kind of three-dimensional looping or some other mechanism.
Yeah, I study snare proteins, and when I started that work in my PhD, and one of the first papers I read was in vitro binding assay, where they said, oh, all of the snares can interact with each other. But biologically they don't, because they're in different compartments or different regions, and that's when I really started getting excited about space
and specificity.
Yes.
And specificity. No, it is a really interesting component of cell biology. So I think the frustration for me is a lot of what I do, because I'm looking at specific DNA sequences, is done on fixed cells. So I lose the dynamic component of you can try and infer it from lots of static pictures. But my dream experiment, I have in the bath on a Sunday night in my head is to be able to watch everything in living cells. And I'm sure it'd be possible. I mean, the pace of change of technology is just so fast. I'm sure it'll come. But I mean, we can watch individual DNA sequences, but only repetitive sequences. We just don't have the sensitivity to see a single gene in a cell nucleus in a living cell. At the moment, so that's the limitation that we're trying to overcome at the moment.
Well, technical limitations.
It's a technical limitation. Absolutely. It's a problem of microscopy and imaging, signal to noise, and quantum yields of fluorochromes, and a whole number of technical issues, which I'm sure will be overcome very soon. So that's going to be really exciting to watch. And then be able to do that, in the context of the organism.
Right.
So moving outside of just cell culture and actually watching things in vivo, which is harder, particularly for the vertebrate. But I mean, my lab has recently started using zebrafish as a very important model organism, because it's a transparent embryo, so it's actually really amenable to live cell imaging. And the other thing we're doing more of now is actually, as a kind of intermediate between cell culture and a real organism, is using organoid cultures. So you can drive.
So what are these?
You can drive stem cells to differentiate into three-dimensional structures. For example, the optic cup. So you can, development again is its self-organizing principle. So if you can get the right cell type started in the culture dish, they will start to organize into three-dimensional structures. Recently published this year, which actually is from a friend of mine who did a postdoc in the same lab as me, was making a kidney in cell culture, starting from stem cells. And also in my institute we do something called cerebral organoids. So you make mini brains from stem cells. So you start to get cell interactions and the right cell levels. It's not quite in vivo, but it's a good intermediate step. And of course it's very amenable for imaging, because it's all just done in dishes.
I mean, I think this idea that you might be able to have kind of replicate organs or tissues in an accessible situation or condition on the bench is a great, and that people have been working towards that for quite some time.
Indeed.
So and
you can test hypotheses. You could use it for screening for small molecules, drugs,
and much better for things like that, because the inherent natural organization in 3 dimensions can sometimes totally change the way that these cells interact.
Absolutely.
I know that that's the case for liver.
Yes.
That 2-dimensional liver doesn't really act like liver.
No. So definitely 3 dimensions are important.
So you mentioned that you like gardening and puzzles. Are these things that you still do now in your free time?
My problem is I have very little free time, unfortunately. I try and get out in the garden every weekend. I do like gardening. My dad was a gardener, so I inherited that from him. I like being outside. I haven't done it for this last year, but I'm quite into tree conservation. So I sit at a desk all day long. So in my free time, it's nice to be outside and doing something physical. Doing physical work is a real pleasure. So I belong to a charity called Trees for Life, which is trying to restore the Caledonian pine forest. So you go up and you live in the Highlands somewhere and get muddy and dig. Which is very therapeutic.
Some physical labour.
It's physical labour. It's being tired, but in a completely different way to being tired from sitting in front of a computer too long. I have two big dogs, so I like going out walking with them. I play tennis rather badly. And the other thing I really like doing is painting and drawing. So I always liked painting and drawing when I was at school, and I kind of gave up when I went to university and lost it somehow. But then I started about probably 5 years ago. I went to the Botanic Gardens in Edinburgh and started doing a botanic illustration course there.
Oh, nice.
And I really loved that. I find it so relaxing. But unfortunately it takes time. So I did for a couple of years. I used to take a Monday morning off work and spend that painting and drawing in the Botanic Gardens, which was nice. But now I can't take a Monday morning off. Monday morning. So I used to save up my holiday time basically and just say, I'll take a half day off every week.
Yeah, I'm sure you know that there's like a big conversation now about work-life balance, and if that is one, possible, and two, how do we encourage that in students as well as investigators. I know that I haven't figured it out.
I think very few of us figure it out, and but I am people say, oh, you have to work hard in science, which you do, but I am, and people in my lab do work hard, not because I force them to, because they want to, but I am a big fan of taking a break, because I think it makes you think clearly. I think if you just keep working, you can't see the wood for the trees anymore. So you really need to step back sometimes. So even if you're doing the gardening, you can be thinking about, how can I do lifestyle imaging while you're doing something with your hands and getting mud all over yourself. So it can be very helpful to step away from it. But work-life balance is an enormous problem now, because now I have 2 jobs in one. I have to do what I did before, running a lab just the same way, and then do a whole new job on top of that. So it becomes almost impossible.
How do you view your time management, or how do you help your students deal with time management? Do you find things that are helpful for you but maybe not for your staff, or vice versa?
I've had to change radically the way I work since I became the director, actually. So when I was a PI, I think I managed the time quite well. So I always tell my lab, I know I'm very busy, but my door is always open, and that you always come first. If you've got a problem, just come and see me. Everything else can wait. And that was true when I was just running a lab, because the lab was the most important. And actually now it can't be. Now actually the institute has to be most important. So now I have no control over my time, because other pressures dictate my timetable. If there's a deadline to return paperwork to the MRC or to the university, or to sign off someone's grant, I have to do that by the date, right? So I'm no longer in control of my own deadline. So that's a difficult thing. So now my lab knows that I can't always respond directly to them. So they've had to improve their time management. And they don't always do it. So people still do come last minute and say, oh God, I need that letter of recommendation for you for this deadline tomorrow. Okay. Why didn't you tell me a week ago?
That's interesting. I think that there's one aspect that we as scientists, I think, talk about is the freedom that we have over our own time. That we may be in the lab for 8 or 10 hours a day, but what we do in those 8 or 10 hours is really kind of up to us.
And it's up to you what those 10 hours are, whether it starts at 10 o'clock or 8 o'clock.
Of course. And that we can take half days to take courses, or go to the gym, or do whatever we want. Same thing with holidays. We can kind of take it depending on how our... we are the masters of our own time, which sounds fantastic, but also you are completely responsible for it. So if you aren't managing your time, or if you don't have your time managed well, then you really aren't very productive. So on one hand, it sounds almost like a relief to have good deadlines, but on the other hand, it's not really how we're used to managing our time either.
Yes, because it's very easy at the end to say, oh, I can stay 1 more hour in the lab to finish the experiment that I haven't started in the correct time.
Yes,
and then basically you are losing your life outside the lab. So it's a fine tuning.
I think we're better at training people to do that now than we ever used to be. I mean, students now, their thesis committees help people plan. When I was a student, nobody ever helped you plan anything, right? I didn't even think we had a thesis committee. I mean, you just were supposed to produce a thesis at some point.
That is good, because students should realize that these panels are not for examining or like to show if you are good or not. It's more like to help you.
Yeah, no, absolutely.
That's I think that there's the idea of evaluation has really changed, even I think from the time that I went to a very progressive undergraduate college where students really had to be in charge of their own education. So and I made that decision because I wanted to get out of the public school system of an A means you're good, a B means you're okay, and anything else is you're not doing very well at all. And I wanted to more understand about my own personal progress. And so evaluation to me was a way of checking in to see how much I knew or how much I was progressing. Whereas now it seems like some sort of endpoint, I take this course, I take this exam, the end. It doesn't seem to be so much of a constant evaluation of how you're doing or how you're progressing over time.
Yeah. Although, I mean, I don't know if it's true in Glasgow, but we have now this kind of annual performance review for everybody in the lab. So there is a time every year where you sit down. Yeah. So every year the postdoc writes down what they aim to do in the next year. Yeah. And then a year later you sit down and go, okay, how much of that did you achieve, and if not, why not? And what can you do better?
We have the same protocols here because I have been in Edinburgh as well, and in Glasgow, so I know the systems are quite similar. Although I have to say that here is a bit more. You have more boxes to fill. That's about a bit more repetitive, but still. I mean, we have to see even for our sides that we are postdocs, and sometimes we think, oh, another review to fill all the boxes, but we have to see the good side.
Yes, the good side. And it's not the paper that's important, it's actually the process, and actually sitting down with your PI and having a conversation, that structured conversation, and with them once a year.
It's kind of self-reflection.
It is,
kind of along the same lines about sitting down with your advisor or your PI. Both of our universities are involved in the Athena Swan scheme. Yep. Do you have any suggestions about how students or postdocs can best take advantage of this type of scheme?
I mean, the fact that it's there, I think, is enormously empowering for them as well, and it just feeds into all the activities of what we do in the university, I think. And we have a postdoc society that regularly has Athena Swan events, so I think they feel empowered to influence things much more. So I think they are taking advantage of it, and organising Women in Science days and things like that. Just because people are talking about it and organising things, I mean, it's having its effect that way, rather than taking advantage of specific modules or courses or anything like that. It's just people's awareness is so much higher.
Do you think that has been a kind of tangible awareness, like in your perspective across your career?
Yeah, very definitely. Yeah. I mean, that idea of you can't have a meeting outside of core hours, or you shouldn't try and have a meeting outside of core hours. I mean, no one even thought of it 5 years ago or something like that. And although we're not perfect, and sometimes you do have to have meetings outside of 10 till 4 or whatever, people are much more aware of it. And actually I find myself kind of speaking to it without even thinking about it. So one of my students said to me the other day, we were out in the pub one evening, he said, we should have a laboratory. And I said, great, let's do that. We haven't had one for 18 months. And he said, and I said, Ilya, you organise it, I haven't got the time. He said, great, let's go skiing, or let's do it. And they wanted to fly off somewhere. And I said, that's great for most people in the lab, but actually, these two people won't be able to go because they've got young children. So actually we need to have a retreat where the people who've
Everybody
got young
can
children,
join.
everyone can be part of it.
Yeah.
You can't exclude people. But,
Nice.
So
Yeah, I was I don't know if I was talking with Maria about this, but or someone else about how I've been very fortunate to work mainly with women. Before I went to graduate school, both of my supervisors were women who were directors of their own research departments. The only male boss that I've had is my current one. So I hear, when I go to meetings about women in science, I hear sometimes horror stories about, not particularly in my generation, but I think in previous generations of, issues that they had in terms of being excluded based on their gender, whether it was where decisions were being made or whether they were invited to things or anything. And I have to say, I very rarely have experienced any of that because I've mostly worked with women.
Yeah. No, it has changed. Yeah, but it's not. I think this one's not just about gender, is it? It's about other things. I think we've also changed the lab culture a little bit. So lab celebrations and Christmas lunches and things like that always used to revolve around consuming large amounts of alcohol. But now we're much more aware that there's people in the lab who can't or won't drink for whatever cultural reason. So actually people are much more aware of, okay, so maybe we should do afternoon tea instead, or something else that doesn't involve going to the pub, which not everybody would be comfortable with.
Yeah, that's true. Yeah, it's kind of not just encouraging diversity, but also acknowledging it. Thinking about it when you organise things. Yeah.
So speaking of celebrations, when do you have any special celebrations that you do if you have a PhD student graduating or a paper?
So for PhD students, we've recently changed actually in the institute to having the students give a public talk in the morning to make the whole day a bit more of a celebration for the institute, and then of course afterwards there's champagne in the labs, and then people just tend to wander down the pub basically.
I think that's an excellent idea.
It's really
to
nice
have
idea.
them give a talk.
This is something that happens all around Europe.
That's where I first experienced it, and I really like that idea.
It's only in UK that they don't do that.
And so we've just adopted it actually.
I also think it's great because it means that you get more experience talking about your work, which as someone who is a very nervous presenter, I always kind of blanch at the idea of having to give a talk, but I always say yes, because it's the only way that you ever maybe not get comfortable with it, but at least get used to it. And I think that more and more people should always have opportunities to talk. So it's awesome to, I think, especially if you've written a paper or you've had a dissertation.
It's also a good opportunity to show your research in the general public. Not only these three reviewers, that
your friends can come who are not
exactly,
scientists,
and your family can celebrate,
come
your
too.
partners,
just makes the whole thing more
people that are very important helping you achieving this point.
So I think more UK universities should start.
And it's good outreach. We have a institute-wide seminar that Maria is part of organizing, and that's nice because as kind of plant biologists, we're part of the whole life sciences, but we very rarely talk to animal scientists, even though we're all, as you said, we're all cell biologists who just happen to work in different systems. So it's really great to kind of learn your neighbors and see them. Because I find oftentimes I get good ideas about something that I could try because someone is doing something completely different, but using maybe similar techniques. And I think, I haven't tried changing that media about that, or maybe I could try.
See, for example, Wendy already said that your, let's say, big dream about where your science goes is of these limitations that you have, technical limitations. So I guess working with physics department or people or optics, more specialized department, then you have to collaborate. So having this kind more general seminars where people from different disciplines attend, then it can give you this.
So I a few years ago I set up something called the Edinburgh Super Resolution Imaging Consortium, between my institute and Heriot Watt University in Edinburgh, which is very good in photonics and optics and physics. So we run a joint PhD studentship and help biologists start to do advanced imaging like that. Good. So and that's been really exciting, kind of trying to bridge that physics biology gap.
Yeah.
Okay.
Cool.
Last
questions?
Yeah, maybe one. You travel a lot, and scientists do travel a lot.
Yeah.
So, and I know that you said already to us that you were in Australia, but what was one of your favorite places that you have visited? Maybe for conference or for holidays?
I think my two favourite places are New Zealand and Hawaii.
Excellent.
So both far away, yes, both in the middle of the ocean, so obviously like islands. I don't know where it comes from, I've got this fascination about volcanoes. I think it's because living in the UK, it's such a geologically old and settled and rather boring.
But you live in Edinburgh, where...
Well, I know it's got a big volcano, but it's long extinct, right? Exactly. And most of the rocks in Scotland are pretty old. I'm fascinated by these kind of violent places where the earth crust is so dynamic.
But not Iceland or Greenland.
No, I do not. It's cold there. I find Iceland very interesting, but it's a bit cold. And I haven't been to Greenland yet. I also like Japan, which is got full of volcanoes, but that's harder work because of the language barrier, I think. But New Zealand and Hawaii are easy. Good. It's really fascinating. Nice. And it's definitely one of the perks of science. You get to go all over the place. Yeah. Amazing. Yeah.
Okay, maybe a last question, and I guess this is also a way that people can find you, is that you have a Twitter account.
I do, yes.
And I guess this is one way to follow, for people interested in you to follow you. But also, do you think social media is something that is emerging in science when it comes to communicating?
Interesting. So I started using social media 2 years ago, probably, persuaded by my colleagues in the institute who are computational biologists or physicists. So I think they've been using social media to communicate for much longer than biologists have. And it also coincided with them persuading me to put my papers on the preprint servers, like the bioarchive. And what they said was, if you put your publications on preprint servers so people can read them before publication, and that's great, but people won't find them unless you tweet about them. So the two things are linked. And actually now, so I find the biomedical literature so enormous, I can't keep up with it anymore. I've given up kind of doing PubMed searches and stuff like that, so I just use Twitter to find interesting papers, so follow groups who are interested in the same things. It's a remarkably effective way of keeping up with the literature in your area, actually.
We say that to me and Emily all the time, that is having your personal journal club.
Yes, indeed.
This is how you treat your Twitter.
Yes, that's what it is. That's what I use it for, almost entirely. I mean, advertising stuff about the institute, but mainly finding papers, or advertising your own papers. Good. Good. So that's to me, that's a revelation, actually.
And you've embraced it. Yeah.
First thing I'm a geek. First thing I wake up every morning, get my iPad out and see what's in my Twitter.
I think that's a way to really reach the generations, through that technology.
And it's also great for keeping up with kind of the social, the science policy side and all the news. Brexit, the Science is Vital campaign in Parliament at the moment. Everything that's going on in America, God help us. And getting scientists' reactions to that, and what you can do about things, and mobilising the science vote and all those things.
And I think that has been very fascinating. Most recently is scientists really going from being neutral position to saying we're not neutral.
Yes,
actually.
That's right.
And that we don't have to be. We were having this conversation at lunch about how scientists, we present information for which decisions can be made, but we as scientists do not make those decisions.
Yes, that's right.
But I think actually that is potentially not.
Now we're starting to give our opinions. Yeah,
that's not the voice of science.
It is. Yeah,
that's required nowadays.
Absolutely not. We have to be much more proactive,
particularly in talking with non-science communities, not just politicians. I'm the only scientist in my family, and so when I talk to my family, I always say, you have to pay your taxes, because if you don't, I don't get paid, and we will never be able to make better electric cars or have better cancer treatment or have food resources that are sustainable. So, you know, every time someone wants to slash your education or your any type of science outreach, but how do you say that to someone who doesn't understand what you do on a day-to-day?
And I'm remarkably encouraged by how receptive politicians are to science, actually. So all my interactions with MSPs or MPs have been really positive. And I think they do understand how important science is, actually. I think they do get it, and I'm not sure 5 years ago so they did. And that really is, just due to the lobbying by all the, you know, Save British Science and all the Science is Vital, Royal Society of Biology, all these Royal Society lobbying groups have just been going on and on about how fantastic universities are at producing knowledge.
Good.
Yeah. Great.
Excellent.
Thanks for spending this time talking with us a little bit.
So we get to know a bit more about Wendy.
You do.
Yeah. Excellent. No, thank you.
Good.
Thank you.
My pleasure.
So hope you enjoy, guys, the conversation. You can check our news once again in our institute's website and all the social media from the institute. And also you can follow us in Twitter. My Twitter is m_Papanatsiou.
And I'm at Larson _PhD.
And of course you can follow Wendy at her own Twitter, so it's Wendy _Bickmore.